Hear from Ziad Rifi. He is a medical student at UCLA. He did his undergrad at the University of California, Berkeley, where he majored in molecular and cellular biology. He was also awarded the Evans Memorial Prize in Physiology for research on the cellular mechanisms of somatosensation, which is a topic we're going to hear about today. During his research year in Dr. Osif Bari's lab, he developed patient-specific connectivity-derived targeting techniques for DBS and focused ultrasound, with the goal of optimizing neuromodulation therapies to alleviate tremor, pain, and depression. He was also granted the prestigious LA Care Scholarship for his international humanitarian efforts and induced into the Gold Humanism Honor Society. Looking forward to hearing from you, Ziad.
Thank you, Dr. Conner. All right, so hi everyone. My name is Ziad Rifi, and I'm a fourth-year medical student at UCLA. Well, before I begin, I just want to thank you all for welcoming this past month. I've really enjoyed my time here, and it was such an honor to work with you all, so thank you. And with that said, today I'm excited to share with you my research on the somatotopic organization of the sensory thalamus and the new insights we've gained from deep brain stimulation for neuropathic pain. So neuropathic pain is pain due to lesions or disease directly involving the central or peripheral nervous system. It affects 25 million adults in the United States, which is over 7% of the general population, and accounts for nearly a quarter of all chronic pain patients. So as you can imagine, a wide variety of treatment options are available for chronic pain, leading to over $600 billion spent on medical management per year. Despite all this, neuropathic pain is often refractory to standard medical therapies and opioid analgesics. Although not yet FDA-approved, deep brain stimulation has been shown to be a highly effective treatment for medication refractory cases. Several targets for DBS have been explored, most commonly the ventral posteromedial and ventral posterolateral nuclei of the thalamus, targeting pain in the face and limbs respectively. But other targets being explored include the periventricular and periaqueductal gray, as well as the anterior cingulate. So the target we most commonly use at UCLA is the VPM or VPL. While these nuclei are clearly visible on histology, they are virtually invisible on MRI. So therefore, surgeons are currently limited to indirect targeting of the VPM and VPL, which necessitates using standardized atlas-based coordinates, using proximity to other landmarks such as the anterior and posterior commissures. For example, the VPM is located anterior to the posterior commissure, about a quarter of the ACPC distance, and 8 millimeters lateral to the wall of the third ventricle, whereas the VPL is 10 millimeters lateral. So while indirect targeting is useful, it doesn't take into account variability in individual anatomy or structural connectivity. This necessitates that we operate on an awake patient in order to perform intraoperative testing to confirm that our lead has been accurately placed. So as this promising treatment option continues to grow in popularity, we need a better understanding of thalamic somatotopy to improve clinical efficacy and outcomes for our patients. So what do we know about thalamic somatotopy? Well, on the left here, you can see the first homunculus theorized by Penfield and Baldry in 1937. Along with Rasmussen, Penfield went on to perform cortical stimulation experiments in about 400 epilepsy patients undergoing awake craniotomy. And with this information, they established the homunculus that we all know today, which shows a medial-lateral hierarchy of cortical body regions proceeding from the foot most medially to the face and tongue laterally, as shown by this figure in the middle. So eventually, Penfield and Jasper hypothesized that the thalamic homunculus mirrors the somatotopy of the cortex, where the tongue and face end up most medially in the VPM, and the arms and legs proceed laterally in the VPL. However, they didn't have access to the direct thalamic testing at this time. So instead, they made this homunculus based off of inferences from animal studies. So we sought to verify the somatotopic distribution of the sensory thalamus using intraoperative macrostimulation from our DBS patients. We were interested in the most clinically relevant body regions, which were the tongue, face, including the trigeminal territories V1, V2, and V3, as well as the arms and legs. With this information, we hope to identify the optimal coordinates for targeting these specific body regions with the goal of personalizing DBS for neuropathic pain. So we started by performing a retrospective review of all neuropathic pain patients who received VPM or VPL DBS at UCLA within the past 10 years. These operations were all performed using the standardized ATLAS coordinates I mentioned earlier. Intraoperative macrostimulation was performed after the initial implantation, and the lead positioning was adjusted until the patient experienced an adequate paresthesia in their affected pain region. We then reconstructed each individual's final DBS electrodes in 3D MNI space using a preoperative T1 MRI and a postoperative CT in a software known as Lead DBS. After this, we identified the MNI coordinates of the center of each contact and assigned them to the primary sensory region perceived during intraoperative testing, and then statistically compared the X, Y, and Z coordinates of these body regions within the sensory thalamus using both a multivariate and a univariate analysis. So here you can see an example of what that output looks like. There's the 3D reconstructed electrodes from a handful of our patients represented in Lead DBS T1 MNI space. In blue, you'll see the VPM, and in pink is the VPL. These are as defined by the Thomas Atlas and Lead DBS. So this was our final patient cohort. We had a total of 14 patients, 10 of whom received VPM DBS for facial pain, and 4 received VPL DBS for limb pain. As you can see, some patients also had a PVG electrode placed. However, all the patients ended up using active contacts within the thalamus only. We derived 56 contacts from 14 electrodes to map sensation in the 4 main body areas, being the face, tongue, arm, and leg. We then used the 33 contacts from the face group to map the sensation in the 3 trigeminal territories. Here we report the mean X, Y, and Z coordinates for each region with standard deviation. On multivariate analysis, we saw overall statistically significant differences in MNI coordinates among the 4 body areas, as well as the 3 trigeminal territories. So on univariate analysis of the body regions, we found these significant differences to be along the X-axis only. A post-hoc analysis confirmed these represented a significant medial lateral distribution of the tongue, face, arm, and leg. This is better demonstrated on imaging. You can see here the mean MNI coordinates for each body region along the axial, coronal, and sagittal planes. Now, specifically focusing on the coronal here, you can see the medial lateral distribution I mentioned. The tongue in red is most medial, followed by the face, arm, and leg in green, which is most lateral. This aligns perfectly with the thalamic homunculus that Penfield proposed, and that medial lateral somatotopy mirrors that of the cortical homunculus that we know. So we next turned our interest to the trigeminal territories, and when we compared the X, Y, and Z coordinates of V1, V2, and V3, we actually found differences this time in both the X and Y axes. So taking a look at the axial image here, we see that V1 in red is located most medially, followed by V2 in blue and V3 in green most lateral. You can also appreciate in both the axial and sagittal planes that V2 in blue is significantly posterior to V1 and V3. So to the best of our knowledge, this is one of the first in-human thalamic mappings of the distinct trigeminal territories. This is also one of the first mentions of any anterior-posterior differences in thalamic somatotopy. So in both the cortex and the thalamus, Penfield's homunculi demonstrate that V1 is closest to the hand area, whereas V3 is the most peripheral. This means that in the thalamus, we would expect V1 to be most lateral and V3 to be most medial, as demonstrated in this image on the left. However, our results show the exact opposite. We found that V1 is actually the most medial and V3 is the most lateral. This is not the first time that conflicts have been found with Penfield's homunculus. Contrary to the traditional Penfield view, the motor and sensory cortical homunculi are patchier and more disjointed than we initially thought. And our medial lateral progression from V1, V2, V3 to arm and leg also fits the descending dermatomal model of somatotopy, which Penfield's homunculi do not abide by. So given all this information, we still cannot invalidate the decades of work that Penfield has done. This is a small retrospective cohort using only macrostimulation and not the more sensitive microstimulation. However, this does show the power of direct thalamic testing, and it's a fascinating indication of what's left to be discovered about human somatosensation. So in summary, we confirm that the somatosensory thalamus displays a medial lateral somatotopy as originally described by Penfield and colleagues. We present the first in-human mapping of individual trigeminal territories, suggesting an inverted facial homunculus in the sensory thalamus. Additionally, we show evidence of AP somatotopy for the first time within the trigeminal territories. Not only does this add to our understanding of the basic science of brain mapping, but we believe that more accurate targeting can also improve the efficiency of DBS surgery, possibly allowing this to be performed in asleep patients, as well as increasing the efficiency of postoperative programming. So in the future, we'd like to perform this intraoperative macrostimulation prospectively with the addition of microstimulation studies. We're also aiming to assess the volumes of tissue activation of our implanted patients to better characterize the clinical efficacy of these targets. Currently, we're working to create a thalamic segmentation map by assessing the structural connectivity of the cortical homunculus to the sensory thalamus using probabilistic tractography. And finally, using all of this information, we can create a somatotopic atlas of the sensory thalamus for the use of operative planning. All of this is with the goal of better understanding thalamic somatotopy to improve body region specific targeting in DBS for neuropathic pain. With that, I would like to thank some amazing people who helped me bring this together, specifically my mentor and PI, Dr. Osif Bari, as well as all the members of the surgical neuromodulation and brain mapping lab. Thank you for your time, and I'm happy to take your questions.
Thanks a lot, Ziad. Any comment from Dr. Capel?
Yeah. First of all, Ziad, really great and interesting study. It's a fascinating topic. You're presumably doing these things at this target to treat neuropathic pain. Neuropathic pain generally of central origin involves a degree of deafferentation. And so have you considered that the somatotopic differences that you're seeing may be due to a reorganization due to the disease process, and that may explain the sort of mismatch that others have looked at in the past, because when they did those mappings, they weren't necessarily done with a disease state?
Yes, absolutely. We know from both cortical and thalamic studies that afferentation of pain and patients with just greater chronicity of pain tend to have larger areas dedicated within their sensory homunculi to those areas of pain. So there is a chance that a patient with V1 pain for many, many years could be skewing the distribution within the thalamus to represent a larger area. Something we're trying to do in these future studies, because we're adding patients every time we have them, is to account for that duration of pain, and also to then prospectively target further lateral, further medial to assess the boundaries of that area. So if a patient does have a V1 distribution of pain covering half of the thalamus, then we can account for that variability.
And just from a bog standard operational standpoint, you are sort of documenting the degree of at least reorganization that is appearing, phenomenologically, correct, I assume?
Yes. And part of the prospective targeting is that we can then, right now we basically can insert the electrode and stimulate until we adequately create a paresthesia that the patient is comfortable with, but we can't test further than that. So what we would like to do is be able to do more prospective testing just to get an assessment of all the body parts within a patient.
Great study.
Thank you.
Dan Cummins?
Yeah, so I have a couple of questions. I guess the first thing is that, from a purely scientific perspective, it's actually kind of an indirect method to map the homunculus based on benefit to stimulation, right? So, perhaps a more precise measure would be evoked potentials to sensory stimulation. So I'm curious if you also have that data and how you think or that does overlap the actual evoked potentials to sensory stimulation in the area that you're stimulating. So that's kind of question one. And then question two is, clinically, one of the issues with neuromodulation of chronic pain is that these patients sometimes have benefit acutely, initially, and then habituate over time. And so, what do you think is happening there? Is it similar to what Dr. Gopal was mentioning, but over time with stimulation, are these patients reorganizing their sensory thalamus, or how do you think that that's happening, and what do you think we could do to potentially overcome that?
Yeah, those are excellent questions. Thank you. So regarding your first question, yes, I do agree that those would be that would be interesting data to see. I think part of why we focused on the intraoperative macro stimulation here is that this is more of a clinically derived question. So how can we best target for those patients who are going to undergo DBS for chronic pain? But it would be very interesting to combine this with the somatosensory evoked potentials and see, do these align still? Do they not? So that's part of why we hope to do the microelectrode studies, and we're applying for that equipment and everything, and hopefully I can have an answer for you soon. The other part of it is, yes, we do see a lot of times with patients who undergo DBS for pain that they eventually kind of plateau in their efficacy and have to undergo some form of cingulotomy. And we don't want that for every single patient. And part of it could be overstimulation due to inaccurate targeting. So if we're constantly overstimulating to reach an area that we haven't properly targeted, then that could also cause some afferentation and deafferentation and shift the efficacy of this. So we hope that if we're able to target off the bat the correct area, we can use lower dosage stimulation, lower frequency and amplitude, and hopefully that can preserve the duration that someone would need until they get a cingulotomy down the line.
See, that was a great talk. Love the consistency of your approach and sophistication. It's interesting, isn't it, that the MRI seems to be blind to these differences? You're using tractography now, but there must be more differentiation on a cellular level than just different tracts. You think 7T or some other type of MRI could differentiate?
Possibly. Actually, something we're using now for VIM DBS for essential tremor is the F-gator sequence within MRI. And with that, we're able to see slightly better differentiation, as well as some automated tractography that goes along with that. So if we could build some sort of protocol to directly build that into the MRI sequencing when we order for patients, that would be such an effective way to differentiate these nuclei.
World of research open to you just for this one profound question. Great.
Thank you, everyone.
All right, thanks a lot, Ziad.